Hello everyone, myself Gayatri. I am from the Department of Zoology, PSG CAS Coimbatore. So in this video we are going to discuss about the nucleus and its development in the embryology point of view. Let's move on to the video. First, these are the contents we are going to look upon in this video. First we are going to see about the enucleation experiment which is done in the nerve cell and sperm cell. And then we are going to see about the experiment on the nuet cell and nuclear transplantation in Astabularia, and finally we are going to conclude this video with the nuclear factors that is involved in the development of the nucleus. So the first one is the enucleation experiment. As the name suggests, enucleation is the removal of the nucleus from the cell. So we are going to see in the upcoming slides how we are going to remove the slide from the sperm cell and the nerve cell, and we are going to insert it into the ovum. So this experiment is carried out by Gordon, and he done that experiment in the year 1968. So the experimental organism which he used for this experiment is the frog's egg. So first one, enucleation of the nerve cell. Nerve cell is nothing but it is a neuron. So when we are removing the nucleus from this neuron, this process is called as the enucleation. So the nucleus from the neuron is inserted into the enucleated ovum of the frog. So this nucleus tend to adapt to the egg, that is the egg cell. So we know that neuron is incapable of dividing, but when this nucleus is inserted into the enucleated ovum, it starts to behave as the nucleus of an egg and it starts dividing. So he found this important point in his experiment. Let's move on to the other one. The second one is the enucleation of the sperm cell. That is, then he took the sperm cell and he removed the nucleus from the sperm cell, which is basically called as the enucleation. So this nucleus which is removed from the sperm cell is inserted into the enucleated ovum of the frog. So then this nucleus behaves like the nucleus of the ovum. From this two, the enucleation of the sperm cell and enucleation of the neuron, we come to the conclusion that nucleus alone doesn't play an important role. Cytoplasm, which is involved, also plays an important role in the development of the organism, and it indirectly influences the nucleus how to behave. So we can take this to experiment under the nucleocytoplasmic interaction, because the cytoplasm also plays an important role in this experiment, that is in the development, in the behavior of the nucleus. So the next one is Siddle experiment, which is done in the year 1932. Siddle took the experimental organism as the dragonfly. So we move on to the experiment. So now the Siddle experiment. You can see the cell I have drawn on your left side. So the green color here denotes the nucleus. It has the two nucleus. The dragonfly cell has two nucleus, and because it undergoes incomplete cleavage, it moves to the opposite sides. That is, one move to the anterior side and the other move to the posterior side. The anterior side nucleus will get divided in the anterior side respectively, and the posterior side nucleus will get divided in the posterior, and it develops into the blastula stage and it develops into the gastrula stage. This is the normal process. What he done is, he inserted a UV irradiated plate in the posterior nucleus and then it got distracted. Now only the anterior nucleus is present in the cell, and this anterior nucleus further divides and it does its same job, that is, it takes care of that anterior part. But what here happens is that anterior nucleus can also capable of inducing the division in the posterior part. That is, it can also translocate to the posterior part, and there also the division occurs. So he concluded that one nucleus alone is capable of development of the normal dragonfly. The two nucleus is not compulsory. So he found out this interpretation in his experiment. So next we are going to see about the somatic cell nuclear transplantation, a very famous experiment. We all know about this. So this was done by Sir Ian Wilmot, and what are the experimental organism he used in this experiment is Pink Dorset sheep and Scottish black-faced sheep. He took this two species of sheep. One is the nucleus donor and the other one is the egg cell donor. We are going to see in the upcoming slide. So what he done in this experiment? So first he took the Scottish black-faced sheep's egg, that is the egg cell. Then he nucleated the egg cell only. Now we have the cell without the nucleus. On the other side, he took a Finn Dorset sheep nucleus. This nucleus is taken from the mammary cell, which is actually called as the udder cell. This nucleus, we know it is a diploid nucleus. So this nucleus is inserted into the cell without nucleus of the Scottish black-faced sheep. And then it develops into the blastula stage. And after the blastula stage, this cell is taken and embedded into the surrogate mother, and finally we can come to know it developed into the dolly, which resembles the nucleus donor here, Finn Dorset sheep. So it resembles like a Finn Dorset sheep. So here is the simple illustration. The nucleus which is removed from the udder cell of the Finn Dorset sheep is inserted into the enucleated egg cell of the black Scottish sheep. So this is done with the use of the electric current, and then it developed into the blastula stage, and during this blastula stage, after the development of the blastula stage, it is embedded in the uterus of the surrogate Eve, and finally it developed into the dolly, which resembles the Finn Dorset sheep, which is a nucleus donor. So from this experiment we can conclude that the nucleus plays a very important role, because the nucleus is taken from the Finn Dorset sheep, and even though it is inserted into the enucleated egg of the other species, it finally resembles the nucleus donor. So from this experiment we can really conclude that nucleus plays an essential role, and nuclear factors plays an essential role in the development of the whole organism. The next one. So now we saw the enucleation experiments. So first was done by the Gurdon and Sir Ian Wilmot, and the other experiment we have seen in the Siddle experiment, which is done in the dragonfly. So these are the three experiments we saw about the enucleation. The next one is the nuclear transplantation in Acetabularia, which is done by the scientist Hammerling. So what he done? He took the two species of the Acetabularia. One is Acetabularia mediterrana and the other one is the Acetabularia crenulata. We need to know that Acetabularia is the unicellular algae, that is, it contains only one cell. But it is a macroorganism, that is, it is 6 cm in length. It can be visible through the naked eye. We can see here, right? The top portion is called as the cap, and the middle portion is called as the stalk, that is, the stem portion is called as the stalk, and the bottom portion is called as the foot or rhizome. This foot or rhizome contains the nucleus. So these are the three parts. Remember, cap, stalk and rhizome, and remember a very important point, the rhizome contains the nucleus. So he took the two species. What he's done? So what Hammerling done? Hammerling at first removed the cap portion along with the stalk of the Acetabularia mediterrana, and it regenerated the, that is, the rhizome regenerated the cap. But the cut portion, that is the cap along with the stalk portion, doesn't regenerate the rhizome, because only the rhizome has the nucleus here, so it regenerated the cap and the stalk portion. But the cap and stalk portion doesn't have the nucleus here, so it cannot regenerate the rhizome. So this is the first concept. This he done in the Acetabular meditraina as well as he done it in the Acetabular crenulata. And the second one is he removed the stalk of the Acetabular meditraina and then he placed it over the rhizome of the Acetabular crenulata. That is, he is basically transplanting this, and it resulted in the intermediate species. He again done it vice versa. That is, he removed the stalk of the Acetabular crenulata and placed it over the rhizome of the Acetabular meditraina. It also resulted in the intermediate species. So, what we can conclude this from this experiment is the presence of nucleus has an important effect on the development of the species. You can see the rhizome has the nucleus, so it can regenerate. The stalk portion and cap portion doesn't have the nucleus, so it cannot regenerate. So the nucleus plays very important role in the development of this algae, which is Acetabularia, and we can also see the intermediate species is also obtained. It should also be noted here. So the third experiment is done on the Nucleus cell. It is done by the two world famous scientists called as the Hans Spemann and Mangold. They done this experiment in the year 1928. They also done the other experiment for which they got the Nobel Prize in the year 1935. So that is also an important experiment. So first we look into this experiment. So what he done? He took the nucleus cell and he tied it with a very thin strand of hair. The nucleus which is present is stuck with the one side of the cell and it continues to divide. It forms the many blastomeres and there would be the stage of the blastula, gastrula, but it happens at a fulfilled stage on one end only, on the one side of the egg only. So what happens in the other side of the egg? So that lacks the proper nucleus, that lacks the proper division. So they resulted in the formation of the two nuet, but the abdomen region is not developed in the one nuet, which tells us about the importance of nucleus, that is the distribution of nucleus throughout the cell is very important. So what he done at the second case? He took a cell, he then again tied a thin strand of hair, but he tied very loosely. So, the nucleus which is stuck at the one end continues to divide and the blastomere from that one side moves on to the other side because he loosely tied the hair. So, it moves on to the other side and as usual the stages occur at the both side of the egg which results in the formation of the two nuets. So, this side also blastula, gastrula stages will occur. This side also blastula, gastrula stages will occur. So, it develops into the two nuet cells, which is not possible in the first case because we tied it very tightly. So there would be no transportation or translocation of the cell from the one side to the other side. So it resulted in the jointed nuet cell which lacks the proper abdomen. So what are the nuclear factors that is involved in the development of the nucleus? We are going to look upon into that. So several nuclear factors are involved from the DNA replication to the formation of the protein. They can be grouped into the genomic level, transcriptional level, post-transcriptional level and translational level. So first at the genomic level, there may be the mutation or DNA methylation can occur which affects the process. And then in the transcriptional level, there may be the lack of RNA polymerase that help in the formation of the mRNA or the DNA in the nucleus may also get produced. That is, it fragments the DNA molecules, so there will be no transcription. Topoisomerase and helicase change the three-dimensional structure of the DNA, so there will be no transcription. So these are the things that take place in the transcriptional level. So then we move on to the post-transcriptional level. Here there may be the masking of the mRNA by the methyl group or inactivation of the ribosome that synthesizes the mRNA or degradation of the mRNA may also take place by the RNase. These comes under the post-transcriptional level. Finally, translational level. Here, the polypeptide which is formed, polypeptide bands which is formed are disintegrated Phosphorylation and glycosylation may also occur, which results in the addition of the phosphate group and carbohydrate group respectively. And lipidation occurs, that is the protein added to the cell membrane, that is plasma membrane, so the cell becomes the non-functional. So these are the things that occur in the different stages. These are the factors that is involved. So that's all about the topic. We saw about the enucleation experiments and nuclear transplantation in Astavlaria, and we saw about the somatic cell nuclear transplantation. Then we saw about the nuid cell, so how the nucleus plays an important role in the nuid cell. So finally we concluded with the nuclear factors, that is in genomic level, transcriptional level, post-transcriptional level, and translational level. So all those things we saw in this video. I hope this video will help you and give insight about the nucleus and its development in the embryonic stages. Thank you so much. Have a nice day.